Hello everyone, welcome to Nutrimentor. This lecture on metabolic health beyond calories, functional nutrition and metabolic health was originally presented as a webinar for IPN India. Given the growing interest in metabolic health and evidence-based nutrition, I wanted to make this content available in recorded format so that students, healthcare professionals, researchers, and nutrition enthusiasts can access it at their convenience. In this session, we will explore why metabolic health extends beyond simple calorie counting and discuss role of functional nutrition in supporting metabolic well-being. Thank you for joining me and let's begin. So when we discuss metabolic disorder, we often default to calories as a primary explanation. But emerging research shows that metabolism is regulated by far more than energy balance alone. Food acts not only as fuel, but also as a biological signal. It continuously interacts with hormones, gut microbiota, inflammatory pathways, circadian rhythms, and cellular metabolism. So when these signaling systems become disrupted, metabolic dysfunction can develop, even when calorie intake appears appropriate on paper. This is why functional nutrition lens is becoming increasingly important in modern metabolic health. It changes how we interpret dietary patterns, how we assess patients, and how we approach long term metabolic regulation in clinical practice. This is a framework most of us were trained in: calories in versus calorie out, weight loss as a primary outcome, and standardized diet chart. And to be clear, this model is not useless. It explains basic energy balance. But where it falls short is in explaining metabolic behavior. We now see that dietary patterns such as DASH and Mediterranean diet improve insulin sensitivity and liver health, even when calorie intake is not aggressively reduced. We also know that two individuals eating the same calories can have different postprandial glycemic and lipid responses. And importantly, timing matters. Chrononutrition research shows that metabolism follows circadian rhythms, not calorie clocks. So calories are measurement tool, but they are not reliable metabolic predictors. Let me translate this into a real world scenario. So this is a clinical pattern many of us encounter routinely. So a case study, a 32-year-old woman with PCOS, insulin resistance, sedentary lifestyle, very sincerely follows a calorie restricted plan, calories reduced, sugar reduced, daily walking added, and yet 3 months later, the metabolic picture barely changes. Minimal weight loss, fatigue persists, fasting insulins remain high, cycles remain irregular. Usually the point where patients say, I followed everything properly. Why is nothing working? And honestly, sometimes they did follow everything properly. The issue may not be compliance. The issue may be that calorie reduction alone does not fully address metabolic signaling because metabolism is not responding only to energy quantity. It is responding to biological information. So the important question becomes what changed when metabolic response finally improved? Interestingly, the breakthrough did not come from further calorie restriction. The shift comes from changing the quality and signaling properties of the diet. Fiber diversity increased, millet, legumes, vegetables included, fermented foods were introduced daily, meal timing became more aligned with circadian rhythm, and food combinations were optimized, especially protein and fiber pairing. Notice something important here. None of these interventions are extreme in nature. No detoxes, no metabolic hacks, no miracle powders, nothing at all. What changed was the biological signaling environment. The shift was the calorie control to metabolic signaling. And once the signaling environment changed, the metabolic response began to change as well. Within the next several weeks, energy levels improved, cycle regularity improved, fasting insulin improved, and interestingly, weight change was modest. But metabolically, the patient was improving, and this is very important clinical insight. Metabolic improvement often precedes significant weight loss, which means if we monitor only body weight, we may completely miss early metabolic recovery. And that is exactly why functional nutrition requires us to look beyond calories alone. So perhaps the better question is no longer how many calories, but rather what signal your food you're eating is sending to your body. Because metabolism doesn't respond to calories alone, it responds to signal. Food sends information to our genes, to our biological clock, and very strongly to our gut microbiota. These signals influence insulin sensitivity, inflammation, and energy partitioning, often before weight changes occur. Let's take a simple example 100 calories from refined sugar versus 100 calories from an apple. Same energy, completely different message. One rapidly spikes glucose and insulin, the other delivers fiber, polyphenols, and microbial substrates, modulating incretin hormones and microbial metabolites. This is what we mean when we say food is metabolic information. When we talk about metabolic disorders, we often focus on individual organs, fatty liver, muscle insulin resistance, or adiposity. But metabolism does not fail in isolation. It fails as a communication network. The gut constantly sends signal, microbial metabolites, and inflammatory mediators that influence adipose tissue behavior, hepatic lipid metabolism, and muscle glucose uptake. Multi-omics studies, including metagenomics and metabolomics, have clearly demonstrated this interconnectedness. So, a disturbance at one node does not stay there, it cascades. For example, gut dysbiosis can increase intestinal permeability, allowing inflammatory molecule to enter circulation. This promotes adipose inflammation, increases free fatty acid release, and ultimately derives hepatic steatosis and systemic insulin resistance. This is why targeting a single organ often gives you only partial or temporary results. If metabolism is communication network, the next logical question is how does food influence this network? Metabolism functions as connected network. The gut produces microbial metabolites, short-chain fatty acids, and inflammatory molecules. These directly influence the liver, altering glucose production and lipid metabolism. The liver then communicates with adipose tissues, modulating fat storage and inflammatory signaling. Adipose tissue releases cytokines and free fatty acid, which finally impacts skeletal muscle, altering insulin sensitivity and glucose uptake. So, what starts in the gut does not stay in the gut, it propagates across the entire metabolic system. This is why we now understand that metabolic disease is a network failure, not an organ specific disorder. And this is exactly why food, which first interacts with the gut, has such powerful system level impact. Metabolism does not operate on a 24-hour calorie counter. It operates on circadian clock. Every major metabolic tissue like liver, muscles, adipose tissue has its own internal clock regulating gene expression. And nutrients themselves act as a zeitgebers, time cues that reset these clocks. This explains why carbohydrate and fat handling varies across the day. Eating pattern misaligned with sleep and activity impairs glucose tolerance and insulin sensitivity, even without excess calories. Importantly, this regulation is tissue specific. Muscle, liver, and adipose do not respond identically to the same timing cue. Emerging evidence from time restricted feeding shows metabolic improvements even without calorie reduction. So, if timing matters, the next question is does the food matrix and combination matters too? So, in nutrition, we often isolate nutrients, vitamin D, iron, and zinc, and treat them as independent actors. But biology does not work that way. Food is consumed as a matrix and nutrients interact during absorption and metabolism. For example, mineral absorption can be competitive. Zinc and copper influence each other. Fat soluble vitamins require dietary fat for optimal absorption. Polyphenols and fiber interact in the gut, enhancing the short chain fatty acid production. A classic example is turmeric and black pepper. Curcumin alone has limited bioavailability, but piperine enhances absorption dramatically, a pharmacokinetic effect. This is why dietary patterns like Mediterranean and DASH diets work so well, not because of single nutrient, but because of synergy built into food combinations. When we combine timing, signaling, and synergy, nutrition becomes a tool to actively modulate metabolism. When we talk about functional nutrition, phytochemicals form one of the strongest links between food and metabolic signaling. These compounds does not act as simple antioxidants. They influence gene expression, inflammatory pathways, and gut-derived metabolites. Polyphenols interact with nuclear receptors involved in lipid and glucose metabolism. Sulfur compounds from cruciferous vegetables influence epigenetic regulation. Carotenoids contribute to metabolic resilience, so these compounds are not passive food components, they are active metabolic regulators. Growing evidence suggests that phytochemical rich diets are associated with improved insulin sensitivity, better glycemic control, reduced liver enzymes in fatty liver disease, and lower cardiometabolic risk of wars. Not all phytochemicals are equally bioavailable. Absorption varies based on food matrix, gut microbiota, and individual biology. So, from the clinical perspective, food-based patterns remains the foundation. When we talk about fermented food, we often reduce them to probiotics. But biochemically, fermented foods are much more than that. Fermentation transforms the food matrix itself, producing bioactive metabolites and modifying polyphenols and fibers. What reaches the gut is not just live microbes, but complex metabolic package. These changes increase microbiota diversity and strengthen the gut barrier and enhance short chain fatty acid production. Controlled dietary trials show improvements in inflammatory markers and insulin sensitivity. In our Indian context, traditional fermented foods like curd, idli and dosa batter, and kanji are practical, accessible examples. The future lies in personalization, understanding dose and individual response, where dietitian can play a major research role. One of the biggest mistakes we make is reducing fiber to a number 25 grams, 30 grams. But evidence now clearly shows fiber diversity matters more than quantity. Different fibers feed different microbes, producing different short chain fatty acids. Butyrate support gut integrity, propionate regulate hepatic glucose metabolism, acetate influence appetite signaling, when fiber diversity increases, glucose control improves, insulin sensitivity improves, and GLP-1 and PYY regulation improves. Emerging indices such as DIGM, that's nothing but dietary index for gut microbiota, show protective effect even before weight loss occurs. So clinically, the goal is not supplements, it is designing diverse plate. I often ask patients just one question. How many different fiber sources did you eat this week? And this often predicts metabolic risk remarkably. Before I conclude, I want to briefly contextualize this for India. Over the last 2 decades, we have seen a major dietary transition from traditional diverse diet to ultra processed refined food. We now consume more polished rice and refined wheat, more packaged foods, and significantly less dietary fiber. ICMR and 9 data from 2024 clearly shows declining diet diversity, reduced pulse and millet intake, fruit and vegetable intake well below the recommendations. This perfectly mirrored our rising burden of obesity, type 2 diabetes, fatty liver disease, and PCOS. What is interesting is that principles we discussed today, like fiber diversity, fermented food, whole food combinations, are deeply embedded in our traditional Indian diets. Curd, idli, dosa batter, kanji, pickled vegetable, pulses, millets, seasonal vegetable. This is functional nutrition in its original form. So, in reality, we are not introducing something new to India. We are scientifically rediscovering what our traditional food system already understood. So, the obvious question is, how do we translate all this mechanistic science into daily practice? The shift is not dramatic, but it is intentional. First, assessment changes. We stop asking how much patients eat and start asking what kind of signal their food is sending. Second intervention, diet design becomes synergistic, not restrictive. Mediterranean and DASH work not because they cut calories, but because they optimize food combinations, timing, and diversity. Third, monitoring improvements. Sometimes metabolic improvements appears in liver enzymes or inflammation before weight loss occurs, and that's critical for patients' motivation. This is where dietitians' role evolves, from giving diet charts to guiding metabolic health through nutritional signaling. And that's the real advantage of functional nutrition. So, if there is one message I would like you all to take from today's session, it, it is this: food is not just calories, food is information. Every meal communicates with hormone, the gut, microbiota, the brain, and metabolic pathways. And sometimes patients don't improve simply because metabolism is more complicated than a calorie counting app. When we shift nutrition counseling from only restriction towards improving metabolic signaling, clinical outcomes often begins to change. And I believe this is where modern clinical nutrition is heading. Have a look towards the references. Thank you everyone for your time and attention. I hope this discussion has given you some useful insights and fresh perspective on the factor that influence metabolic health beyond simply counting calories. If you found this session helpful, please consider subscribing to Nutrimentor for more content on nutrition, research, health, and academic learning. Thank you once again. I look forward to connecting with you in future sessions. Take care and stay healthy.